First of all, I would like to thank the International Myeloma Foundation for asking me to run through some key aspects of the late-breaking abstract that we presented at ASH 2025. As you can see from this first slide, we presented results of MRD negative outcomes following a novel in vivo gene therapy generating anti-BCMA CAR T cells in patients with relapsed refractory myeloma. And this was preliminary results from the inMMyCAR study, which was a first-in-human phase 1 study of KLN-1010. So the first slide here indicates what we did as opposed to what is now the standard of care. And as we all know, the application of ex vivo CAR T has led to tremendous advancements in the therapy of myeloma. And this is illustrated on the left. We collect T cells, then perform gene therapy, expand the cells, and it also involves preparative chemotherapy, followed by the infusion of CAR T cells. What we have done in this trial is to try to expand the reach of CAR T cells using in vivo gene therapy by using lentiviral particles to achieve in vivo generation of CAR T cells. And as you will see, this eliminates the need for preconditioning lymphodepletion chemotherapy. It simplifies the logistics, which helps to expand the access of CAR T therapy. And the absence of the need for ex vivo culture of T cells may also increase T cell fitness. So KLN-1010, which is the investigational product, is a modified replication incompetent and self-inactivating lentiviral vector with a modified envelope containing two synthetic envelope proteins. First of all, a D-targeting mutation was introduced into the vesicular stomatitis viral glycoprotein, or VSVG, which inhibits binding to the LDL receptor, which is the classical mechanism for mediating cellular entry in lentiviral gene therapy, while preserving fusogenic activity and maintaining a high efficiency of transduction. Secondly, the viral vector is precisely targeted to the T cells using a anti-CD3 single-chain variable fragment. These modified envelope proteins therefore target the CD3 positive T cells to enable entry of KLN-1010, delivering a transgene that is encoding a fully human anti-BCMA chimeric antigen receptor, shown on the slide here, which was selected based on high levels of activity to BCMA positive tumors. So the trial that we performed is, as we illustrate on this slide, it's called inMMyCAR, is a phase 1 first-in-human study whereby patients with relapsed and refractory myeloma after 3 or more prior lines of therapy, including proteasome inhibitor, immunomodulatory drugs, and anti-CD38 antibody with good performance status and adequate bone marrow and organ function, were treated with KLN-1010. And the design was the standard 3+3, aiming for approximately 20 patients in the escalation phase, followed by an expansion cohort at the recommended phase 2 dose. And our primary endpoints were to determine the safety and tolerability and the recommended phase 2 dose, while secondary endpoints were to assess CAR T cell expansion and persistence, the overall response, the assessment of minimal residual disease, the duration of response, and progression-free survival. This trial was commenced in 3 centers in Australia, and we reported the results in December so far for the first 4 patients, 3 who were treated at dose level 1 and 1 patient at dose level -1. And the reason we moved to dose level -1 was to explore the therapeutic window because we had found high levels of activity with a favorable tolerability profile, which I will explain in a moment. So as you can see here, the baseline characteristics of the initial 4 patients are summarized here with ages of between 61 and 72. All 4 patients had high-risk cytogenetics, including deletion 17p, amplification 1q, and deletion 1p. They had received 3 to 5 prior lines of therapy. All had received prior autologous transplant with prior PI, IMiDs, and anti-CD38 antibody. In addition, it's important to note that none had prior exposure to BCMA targeting agents, while 3 of the 4 patients were triple-class refractory. But we certainly hope after the initial phase that we will consider patients who have prior exposure to BCMA targeting agents. So because we wanted in vivo transfection, meaning that the CAR Ts are made in the patients, obviously we did not want to deplete lymphocytes. And here you can see a very key finding that despite the absence of lymphodepletion, we were able to demonstrate CAR T expansion. So on the left, all 4 patients experienced a rise in lymphocyte count, with 1 patient at dose level 1 rising to 43 times 10 to the 9 per liter. Dexamethasone was administered to this patient as well as 1 other patient with prompt resolution of the lymphocyte expansion and no clinical sequelae. On the right, you can see that the CAR-positive T cells were detected at day 15 at levels of between 22 and 85% of CD3 positive T cells, with a mixture of CD4 and CD8 cells of different proportions. And of note, the highest level of 85% CAR was achieved at dose level -1. We know from ex vivo CAR T cells that memory T cells are associated with CAR T persistence and durable myeloma regression. So here we show phenotypically that in all 4 patients on the right-hand side of the slide, that the KLN-1010 generated CAR Ts were enriched with these less differentiated and memory CAR T cells, which were a very pleasing finding because we know from ex vivo CAR T, as I mentioned, that it is associated with improved expansion, CAR T persistence, as well as tumor control. And on the graph on the left, you can also see that from plotting of the CAR T cells over time, that the peaks were at day 15, but importantly, that in our first 2 patients, that these CAR T cells persisted to month 3. They are also persistent in the bone marrow with the same memory phenotype. And here you can see the clinical results where all patients have achieved MRD negativity by month 1 to the level of 10 to the -6 in the first 3 patients, and due to sample cellularity to 10 to the -5 in patient 4, where we are waiting sequencing results, with our first 2 patients remaining MRD negative at month 3, with patient 2 again at 10 to the -5, again due to sample cellularity. The IMWG responses are complete response for the first patient at month 5. At the time in December, we had only received the result 1 week prior to that. And so we were awaiting confirmation, and I can say that this has now been confirmed. The others are currently in PR, which likely relate to clearance half-life of the paraprotein, in the setting that all of the patients, as you can see on the bottom left, have serum-free light chains that involve serum-free light chains that have decreased to subnormal or non-detectable levels, as we often see in ex vivo CAR T. And concurrently, as you can see on the bottom right, in 3 patients for whom we have data on soluble BCMA, they have markedly reduced, consistent with the reduction in tumor load and in the other parameters that I've shown you. And importantly, looking at safety, here you can see that cytokine release syndrome occurred in 3 patients, but they were all mild at grades 1 and 2, with no grade 3 or above CRS. In addition, we can also see that there were no ICANS, and to date, no evidence of delayed neurotoxicity. I might also mention that for the CRS, they occurred at days 10 to 12 at the onset, with a duration of 2 to 8 days, and all of them resolved with dexamethasone and tocilizumab. Other aspects of safety are illustrated here, where the toxicity profile was also found to be very favorable. In particular, there were minimal events of cytopenia. The only grade 4 cytopenia was 1 case of grade 4 neutropenia, which occurred shortly after the infusion and which resolved completely within 16 hours, likely reflecting neutrophil margination. All grade 3 cytopenias occurred at around days 14 to 16 and resolved promptly. There was one grade 3 infusion reaction with 2 others of lower grades. They all resolved with tocilizumab and steroid at dose level 1, and the infusion reaction at dose level -1 only required paracetamol. The adverse events of grade 3 with the other profiles shown on the bottom left were all manageable and promptly resolved. For example, there was lymphopenia, lymphocytosis, a case of pneumonia, and also a transient syncope episode, which was unrelated. And as I mentioned before, it was due to this favorable toxicity profile, but also high efficacy that we are now treating patients at dose level -1 to explore the impact. So in conclusion, in this phase 1 first-in-human trial of in vivo CAR-T KLN-1010, we showed that lymphodepletion is not required for in vivo CAR-T cell generation and expansion in the peripheral blood. CAR-T cell expansion peaked around day 15, and the memory phenotype T cells persisted in the bone marrow and blood through month 3. We know that similar outcomes have been associated with durable remissions in ex vivo CAR-T and myeloma. We showed favorable safety and tolerability profiles using this off-the-shelf product, which make outpatient therapy feasible, although obviously more experience as we go further in this trial will enable us to assess the ways in which we should use this drug. The CRS was consistent with those seen with ex vivo CAR-T therapies, and in particular, the cytopenias were notably limited, which is a very favorable finding. We achieved 100% early MRD negative responses with deepening of the IMWG response over time. These MRD negative bone marrow responses were sustained through 3 months in the first 2 patients with the longest follow-up. And while initial MRD negative response and persistent CAR-T cells are prognostic of ongoing clinical responses, it is clearly a priority for us to establish the durability of response in continued follow-up, and updated results will be presented at future meetings. So finally, we'd like to thank all the patients, their families, and caregivers for their contributions to this ongoing study at all 3 studied locations, as well as our collaborators. Thank you very much to the IMF for inviting me, and I hope this webinar has been helpful to the audience. Thank you.